When it comes to rejuvenation-based therapies, most are focused on epigenetic reprogramming, but upstream of that, bioelectrical stimulation induces epigenetic regulators, thereby leading to rejuvenation and lifespan extension. In support of this approach, doctors Ayelet Voskobonic and Yos Dahman did exactly that in their recently published paper, and today they'll present those data followed by Q&A. So welcome Ayelet and Yos, and let's get into the presentation.
Hello, nice meeting you.
Yep, great meeting you.
Hello. So my name is Ayelet Voskobonic, and I'm a professor in Stanford University in the biology department, down in Hopkins Marine Station, which is located in Monterey. And we will tell you today, me and Jas Domen, about a study that we did on the colonial chordate Butrellus schlosseri. So this is how these colonies look. It is our closest invertebrate relative. If we are looking on them, they live across many oceans and seas, from the Mediterranean, the Pacific Ocean, the Atlantic Ocean, and many other places around the world. They live in colonies that started their life from a larvae, a chordate larvae, that settle down and become an invertebrate-like. So here we are looking on a grown-up colony, adult. This one is about 20 years old, so they can really live, they really have a long lifespan. Every petal is actually an individual, and they are producing these colonies through a budding by cloning themselves. So if we sequenced their genome in 2013, and we found that about 75% of human proteins have a significant, based on sequence, proteins in the Butrellus colonies. If we are looking on the tree of life, here are chordates, and here are, which are including those invertebrates species. So chordates, anyone that will have chordata, that will have a spine practically. And they belong to tunicates, which are the sister group, the closest sister group to vertebrate. They start their life as a larvae that was developed inside the mother colonies. It took it about 1 week to create from a fertilized egg a larvae with a nerve cord, an notochord, segmented muscles. This is why they belong to chordate. Then they will settle down, first with just one clone, one individual. So we can see here this oozoid, we call them, because they came from an oocyte. We can see here that they have a central nervous system, they have a heart, they have a digestive system, they have blood vessels, which are here in the periphery, like little fingers. And they already have a bud. This bud will grow, and if one has more than one, they will create those beautiful colonies. So every week, every individual in the colony, so here we are also looking on a colony following its settlement, the adults, the buds, these buds will grow and develop, and at the end of the week, all the adults in the colonies will die through apoptosis and programmed cell removal and will be replaced by their clones. Our studies in the lab show that all this budding is mediated by stem cells, most likely tissue-specific adult stem cells. And because every week the adults are removed, are dying, are getting eliminated, at the end, in these colonies, aging is practically stem cell aging. So all the organs within Botrylus colonies are eliminated each week. The colony's stem cells migrate to the bats, regenerating new organs, and they are doing it again and again throughout their life. And as I showed you before, we are growing here in Hawkins Marine Station colonies for more than 20 years, still came from the same clone. And one of the questions is, do they age? If it's just stem cells that are getting moving from one generation to the other. And they definitely do. And I want to show you here a movie that compare in this size. The blue colony is 1-year-old. This one is 20 years old, this 944, a clone that we were using in this study that we will tell you about. And look about their weekly cycle, side by side. We can see that the old colony is going through those blastogenic or budding cycles in a very precise way. However, please note that we are taking the video with the same scale. The young one is producing much larger individual compared to the old one. So this is the first phenotype that immediately we can see. The other phenotype that is for someone that is studying Botrylus for many years, this one is very robust, it's more unite. You can see that this colony is a little bit more active, everything is more synchronized. Their blood vessels look much better, they have less pigmentation, we can actually see through the transparent body. The older ones, you can see that amply, the blood vessels are looking different, they have more pigmentation, they're a little bit less synchronized, and the takeover stage, when the young generation is replacing the new one, is taking a little longer compared to the young. So let's look on some of the phenotype. One of them is, of course, the change in the size of the individual. So again, here we are looking on young colony versus old one. Both of them were taken using the same magnification, the same scale. And you can immediately, so we traced here the zoids, you can immediately see that this 944, our 20 years old, is about at least 2/3, if not less, compared to the young ones. Here we are focusing on one individual. From the back, we can see their endostyle, their heart, their digestive system, the bats in the size. This is the old one. We trace the endostyle, and you can see how this organ is smaller compared to the one in the young. The same is for the digestive system that is down here. We'll trace it here, and this is the little heart, so you can see that each organ is actually getting reduced. Looking on their central nervous system, so this is their brain. The brain in the adult is going through changes in the number of the neuron that is accumulating, and practically in the middle of the cycle on day 4, they have the maximum number of neurons. In young colonies, it's about 1,000 neurons. In this 15 years old colony that we were compared, it's going down to around 800. But not only the number, also the genes that are expressed in the brain are different. And here we are looking on 200 genes that are associated with neurodegenerative diseases, which are differentially expressed in the young, which are in these two lanes, versus, these are 5 months olds, these are the young, and those are the olds. And we can see that many of those genes, including APP and gene that, based on their sequences, we can actually predict early set-off of Alzheimer's disease, etc., are differentially expressed in those, are highly expressed in those aged brains. Another study that we did is on their clock And here we are looking on the expression around the clock of genes that are considered the master of the clock, like clock and BMALs. And we can see that in the mid-age, so still very, very young colonies, it's they have the circadian rhythm, the expression high during the night and low during the day. In very, very young, they still didn't have their clock. They need to establish their clock. So those genes are actually expressed around the clock. And in the old colonies, they're actually again losing their clock. So the expression of those genes again is not as synchronized and have this nice circadian rhythm that the relatively young colonies have. We can look on their heart circulation. So here is the heart of and the blood flow in those hearts in the colonies, and we can actually count how many beats they have per minute. And here we are looking on every 30 seconds. Young colonies have more robust and nice heart flow. Mid are going a little bit less, but still differentially expressed between night and day. Much like us, when we are going to sleep, our hearts are slowing its rhythm. But the old colonies doesn't keep this rhythm, so there are not significant differences between the night and the day. Yas came with his daughter to visit us in the summer for her that she interned in the lab and did a summer here. And he is a scientist too, of course. And he was looking on they were looking on the hearts beatings in those colonies. And they were asking me, what do you think will happen if we will pace those colonies or try to use pacemakers to increase the heart rate in those colonies? And I told them, wow, that's a great idea. Let's try to do it and let's see what will happen. And I will ask Yas now to continue.
Okay, yeah, so this setup is shown here. So yes, the original idea, we did happen to have a pacer that's basically used surgically in heart surgery for temporary pacing. Obviously not something you would implant. And we played with it with ES cells several years ago. But we thought it might be a good idea to see if we could synchronize or increase the rate of the hearts beating in these animals. And the setup that we eventually came down with after some permutations is kind of shown here. The pacing device is this thing, a Medtronic device. With several wires that come out of it. And in this case, only the tip here shows the actual wire-free. Wire here is covered, but over here it's metal, and here metal again for the plus and minus, and in between is a colony. You can see the orange here. And that allows us to give them stimulation with a defined number of beats per minute and a defined milliampere output. So that's what we tried to see if we could affect their heart rate. And so what we saw eventually, so we started off with actually a colony that was in bad shape, not a very old one, but one that wasn't very happy. because it wasn't quite clear that we would be getting anything. And the idea is we're not wasting a colony if we use that. And we played with that for several hours, doing different things. Put it back up, and then a day or so later, we were looking at it. And I think it was Eilert first looking at it, saying this colony looks a whole lot better than it used to look. And there was a significant improvement. Similar, this is not that first colony, but similar to that colony. This is a very old colony. One of the things we tried later. So this one is has all these old phenotypes that I just described. Small zooids, dense, packed with cells. After, in this case, we treated this colony, we didn't treat this colony. And over time, over about a 200-day period, we see that this one doesn't really change, but that the treated one increases the zooid size again, kind of starting to look like a young colony. And there's some other things that change as well. All the things that you see on the edge of the colony called ampullae, which is the end of the blood vessels, they elongate and they push out, which they don't do in the untreated one. And we see this effects lasted about 200 days. So if you go to the next slide, we see some of the effects on the younger animals, so 6 to 12 months in culture. We see that the heart rate increases after treatment, not to the point we stimulated them at about 150 beats per minute. They go up, but not to that very high rate. Normal heart rate is more in the 60 to 90 beats per minute for these animals. And again, that is both shortly after stimulation and even a couple of weeks later. If we look at the number of buds that are generated and as a result, the number of zooids that are growing, we see that they expand faster following treatment compared to the untreated colonies. Interesting what we also saw, and so these initial experiments were done actually not even in the summer, but in autumn, which is when these animals typically don't form gonads anymore. But we saw in the ones that were treated that most of the animals were still forming gonads, 7 out of 9 compared to 1 out of 9 for the controls. And interesting as well, which we saw a year later, without specifically following the colonies, but when we went back the next summer and tried to determine how many of these colonies were left, we saw that of the control animals, 2 out of 12 were still present, whereas for the PEC treated, the pulsatile electrical current treated colonies, 9 out of 12 were still present and growing and alive. So that suggests a significant increase. If we look at zooid size, again, we see before treatment, there's really no difference between the controls here set at arbitrarily at 100% and the treated ones. But 2 weeks later here, we see that the untreated ones are still set for 100, but that the treated ones now are bigger. And similarly, less clear, but we see the same thing. We see ampullae lengthen, so the length-width ratio increases. shown in these two plots compared to the before treatment. And then we also looked by flow cytometry, which is a technique where the cells, the animals are meshed up into individual cells, and these cells are run through a so-called cell sorter, a machine that looks at every cell and looks at a number of parameters. Typically, that would be staining with monoclonal antibodies, which weren't available for these animals at this point in time. There's now a bunch that have been generated and that need to be characterized further, but that's going to be improving. So this was basically separated based on size and internal structure. And this is based on an earlier publication from the lab, where all these different populations were characterized. So in this case, further out here means bigger, higher up here means mostly a more internal structure. And we can recognize a number of different populations, but they're all fairly large populations. For stem cells, they're typically very rare cells, in the order of maybe 1 per 100,000 cells or so in the mammalian bone marrow. We don't know how many there are here, but so we're not looking at stem cell populations, but we're looking at larger populations. We do see most of them do not differ between old and young, but some do, like these here. Not massively, but again, that is to be expected based on the limitations of the analysis, which, like I say, it's size and it's staining with one particular enzyme marker. Similar data were obtained with old animals. I've shown this picture. In this case, we don't see a massive change in the ampullae before and after. But if we look at the size of the animals, we see that before treatment, there's no difference in size between the colonies. However, between the different time points of the colonies. Whereas after treatment, so immediately after, forget this was 30 days and 200 days, we see a significant increase. And this is a bigger increase than we saw in the young animals, which again is kind of as expected since these animals start much lower. So these were the ones that have been in culture for about 24 years when these experiments were done, 2024. And this is a similar old colony. This one has been in culture for about 14 years, not quite as long as these guys. But we see the same difference. So we see a faster increase in the number of zooids compared to untreated. We see increased size after treatment, again, compared to untreated. And we see some differences between different populations in using flow cytometry. And I don't know what we have. Yeah, and I think that's where Violet.
Okay, so another thing that we did, thank you, Dar. Yes. Another thing that we did is we took the animals for sequencing, for bulk sequencing, the whole system. We don't have here the resolution of specific cells. That probably will be in our future studies. But we wanted to see what is happening on the global, what is the global pictures. So in this graph, we are looking on the young colonies. From them, we have big numbers, so we can really do a good statistic and sequence many of them. And so blue is upregulated, the whitish vanilla is downregulation. The size of the circle is the number of genes that are up or down in those specific pathways. And what we saw is that after 2 hours, 2 hours following the pacing, there is a differential expression between control, one that were not treated, versus the one that were treated. And there is a huge downregulation across all those pathways, kind of a shutdown, kind of a reboot, we call it, immediately after the treatment. Then the next time point that we took is 24 hours after the treatment. And here we are actually seeing the opposite effect. So many genes on those pathways, which include many transcription factors and many epigenetic modulator, signaling regulation, mitochondrial genes that are associated with metabolism, with cytoskeletal, with ECM structures, with immune, all of them are, again, many of them are upregulated in the treated colonies. And this is going back, is kind of maintained. Here we are looking on samples that were taken 13 days and 49 days later, and 132 days later. On this side, we have samples from the old colonies, 24 hours. In this case, from the old ones, we don't have as many, so we were a little bit more conserved in the time point that we could sample them. We don't have the 2 hours point. We will now probably repeat it and see if they also were behaving in the same way, but at least in 24 hours, we definitely see upregulation of many of the genes compared to the control, which is going back to maybe more normal situation in colonies that were sampled 41 and 96 days later. Another phenomena that came up from this analysis, when people are studying human and mammalian and mice following exercise, they see a signature that is seen in an immediate time point, which is few hours following the exercise, versus longer time point. And they see changes in genes that are associated with inflammation, a signature of macrophages, versus regeneration signature of macrophages. So macrophages can be in two different stages, M1 and M2, or in the transition between the two. And those graphs practically showing across many experimental or human samples that were taken immediately after exercise or in a longer time after. So again, inflammation at the beginning and more regenerative state later on. And the pacing Botrytis in the sample that we took a few hours later, we saw the same thing, that the inflammation signature is going up compared to the one like 24 hours later that moved, or even later than that, that moved to the more regenerative size, which suggests a little bit of mirroring or something that is resembled to what is happening during exercise. So those were our results. And in summary, um, we have shown that PEC, the pulsed electric current in Botrytis, causes for a reversion into a younger phenotype, as indicated by the improved growth rate, improved gonadial development, improved morphology, extended lifespan. It suggests alteration in cell population more than 1 month following the treatment, and also the global transcriptomic of a reboot and rebound following a PC. Yes, do you want to take this summary?
Sure. This is just some of the questions that come up, whether a similar approach like the low-level pulsatile current can help stress animals, for instance, due to higher water temperatures. And a similar approach has been advocated for improving coral growth. There are a number of questions that come up and that we haven't answered yet. Like, for instance, is it necessary for the simulation to be pulsatile compared to just continuous? And of course, the other big question that comes up, whether this can be beneficial in mammals and for mammals reach humans. Could it result in some rejuvenation, maybe not of whole individuals, but specific populations? Could it improve fertility? All questions that we don't have any answer to as of yet, but that will be interesting to raise and look into.
So, of course, you can read about this paper in the PNAS. And of course, we would like to, and there are some reports from Stanford about this. And of course, we want to thank all the people that contribute to this work. Tom Levy, Kiara, Katie, Carla, Yas, of course, and Erica, and Kim Gandhi, the group of Steve Quake, Norman Neff from the BioHub that they help us with all the sequencing, Debashi Saho from UC San Diego, Yotam and Aiden that work with us on the clock and also did the analysis of the transcriptome, and for you for listening, and we will happy to take questions.
Thanks, Ayelet and Yas. That was great. So yeah, that was great. My first question, I guess, would be, is this already existent somewhat in humans for people who are on a pacemaker, where that's restoring the electrical activity of the heart, and then they have a better health span because of that? So is some of this already being done only in basically quote unquote sick populations, human populations?
Yeah, pacemaker is a little bit different since it paces when the heart doesn't, right? Whereas this is continuous stimulation, and we didn't even talk about the details, but it's actually 3 times 5 minutes, so it's a fairly minimal stimulation that we ended up with, and we've done various things from overnight to multiple sets of short stimulations. But currently, what we do is 3 times 5 minutes with 15-minute intervals in between. But so it's a different kind, and it is something that we need to look further into, how important it is to do it pulsatile, how important it is. Can you do it longer periods? Can you do it shorter periods? So there's a lot to be determined. But I think there are some differences between this and pacing in a heart. There are, of course, human experiments where people are stimulating with currents, and those will need to be looked into.
In terms of the dose, so how did you settle on pulsatile? And is the pulsatile, you said, it's intermittent versus continuous. So 3 times 5 minutes on and then off, 15
Right.
minutes. So is that continuous throughout the whole day, or that's one singular dose?
That's once.
And then how often? So what was the frequency of the dose? Just once and then up to the...
Once, and then they're left to go for, we see effects for 4 months out or so.
Wow, that's amazing. All right, wait. So digging further into those details, did you have some issues early on with the strength of the dose? So, electric current has a frequency. So what was the frequency that went along with that dose? And then, so I'm guessing there's going to be a U-shaped curve where no dose is the worst, right? And then you get to that optimal dose, which is obvious what you guys saw, or close to that. And then too strong of a dose, too high of a frequency. Now it's maybe too much of a stressor, and then they die faster, or whatever the phenotype may be. So was there some growing pains were there some growing pains in the beginning for
Like I said, we did try various things. Strength-wise, we're basically at the max this machine can do, so we might be able to go up further uh, uh, than we have been doing, and for that we'll need different equipment. But we did see the effect with these, so that's why we decided to investigate in more detail what was happening here. And like I say, we did try different mechanisms. We did ranging from overnight continuous pulsatile stimulation.
Yeah,
Which
I will add in that Kim Gandhi,
Mhm.
that was part, of course, of this study, is a heart surgeon, and we use the same protocol that is used during heart surgeries to bring up, I believe, the heart following an open heart surgery. So we started with this protocol. So our heart beat about 60 when we are in rest, about 60 beats
Mhm.
per minute. And in a surprising way, the Butrillus is very similar. So when we told Kim that, she said, "Okay, so let's go with the regular, what we are using in the hospital, right? Let's start with that." And we started using the same setup. We adjusted the time point. We exposed one of the colonies during the whole night, and we saw that they are totally fine with it. They didn't look stressed. They look good, but they didn't look so much better compared to the kind of around the same thing. So we felt, okay, let's minimize the treatment instead of exposing them during the whole night. And we ended up with those three time points. 5 minutes, 15 minutes break, 5 minutes, and another one, and this is it. The nice thing about it, for example, for this 944 that lived for so many years, when it's falling down and we can see it immediately, they are starting to produce zoids that are smaller. We are coming back and treating them, and we are getting the effect again. So it says kind of we can continue to do it as needed.
Fantastic. So that going back to the frequency question in hertz, like if you can convert that into a hertz of exposing the botrytis to, what would that translate to?
I haven't looked at it in hertz, but it's about 150 beats per minute, the stimulation, which is, what is it, 2 or something?
2 hertz. And the reason I raise that is I wonder if this in people, in addition to potentially cardiac rejuvenation, so the brain operates at alpha, beta, delta, right? So
Mm.
I wonder if exposure, not just the heart, but I mean, if you put a human in basically a self-enclosed device, kind of like an HBOT, hyperbaric oxygen therapy, which is kind of what you did to the Petrillus, only using frequency instead of, oxygen pressure. I wonder if mimicking the frequency changes that change during aging in the brain and maybe rejuvenating that to a more youthful, state. I wonder if that would have a similar effect. What do you guys think?
We so in our studies we are aiming for the brain next. So now we are actually conducting the same treatments with a big focus on the brain, on how it responds, on how it will affect its population. So we characterize on a singular level the what kind of cells do they have in their brains. We did find good candidate for neural stem cells and progenitors. We want to see the effect on those cells. So this is exactly is where our, at least on the Botril side, where our studies are going. People are using, so in the many physical therapists, many psychologists, psychiatrists, people are using in the fields all kind of electric signals and treatments. And people will say that it's improving, none of those were done in a very, or it was done to this level or another, but kind of to really zoom in, to really look on the molecular, cellular, epigenetic, mitochondria. Maybe we are just, you know, inserting some, boosting up the energy that the cells have. So Butrilus is a great model for that because we can play with it. We can look on cellular, molecular levels and really get the mechanism behind what we did in a more detailed way. And then we will be a little more, we will have more knowledge and we will understand better how we can really translate it to the bench,
Yeah,
to
definitely.
the
That's
to the
people.
treatment, not the bench. Yes.
So going back to the heart too. So resting heart rate in people peaks around 50 to 60 years. It actually goes up during aging, and then it starts to come down where the heart gets weaker, and then it's just less beats per minute as a part of that phenotype. So I wonder if the heart would be the first place to start, where starting with that population of people that have a declining resting heart rate would be the first group. I mean, outside of, heart transplant patients, obviously, right? And just in terms of translation. But then the other side of that too is elite athletes have resting heart rates that are very low, that look quote
Mm.
unquote aged, but it's actually the opposite, right? So you have the aged phenotype where someone's got a weak heart, a resting heart rate of say 50 or 40 even. And that's actually probably close to death. Whereas an elite athlete can have a resting heart rate of 40 and it's actually good. I mean, the heart is as strong as it can possibly be. So how would, in that situation, it's easy to rejuvenate the aged heart using a method similar to this, but for the elite athlete, would you have to wait for that heart to get even weaker? I mean, it probably wouldn't be an effect on someone who's already healthy. You've got to actually wait for the aging to kick in before doing the intervention. So what are your thoughts?
Yeah, I would think you're right about that. You probably don't want to start stimulating an elite athlete that probably isn't going to have a whole lot of change associated with it. Uh, and yes, heart rate does differ a lot, and it actually starts really high in newborns, much higher than the 60 to 90, which is really what this equipment is set up for congenital surgery, heart surgery. Um, but yeah, I would agree. And we obviously haven't looked in humans with this treatment, but um, also it would be dependent on how you would do. You really want to do the surgery to get the leads in. There's going to be some limitations in what you want to do in a human. So I would think that you certainly wouldn't be doing it in very healthy athletes, but you would be more starting in people that where things aren't going as they should.
So they would have to, the electrodes would have to be directly attached to the person, just like in the Batrilis. It can't be where the environment is completely sealed off and you're exposing that internal environment with the person in there too to the specific frequency and current. I mean, basically it's indirect electrodes. The whole environment would be at a specific frequency.
Yeah, that would be a different approach. But if you specifically want to stimulate the heart, you're probably going to need the wires closer to that, and you probably don't do that from the outside, but you have to be internal to get it limited to that organ.
Got it. And did you measure heart rate variability, right? It's not just how many beats per minute, but heart rate variability declines during aging. The variability in time in between beats. So I'm guessing for Bacillus, it declines during aging too, just like in people. Did you measure that?
Yeah,
you did that,
yeah,
yeah.
I saw I showed it in one of the so what we saw in the older one that in general they have a lower heartbeat, but also not that many differences between night and day. Of course, those were done, in one point midnight. We didn't go through the whole. We are actually now taking time lapse and video, not time lapse, video imaging every few hours around the clock to measure it in a little bit more comprehensive way.
Right, not heart rate. I saw the heart rate data, but the variability in time in between heartbeats, that's known
Ah,
as
okay.
variability. And that declines during aging, right? Because when you hear 60 beats per minute, we assume that that's exactly 1 second per heartbeat, but that's not true. There's
Yeah.
variability in
Mm.
that time, and especially during aging, that variability declines. The reason I raise this issue is because there are people like me who are tracking heart rate variability and resting heart rate. So if rejuvenating the heart in Petrillus with pulsatile electric current is a part, not if, but that it's a part of that story in restoring cardiac function, systemic rejuvenation, rebooting, gene expression, all of that good stuff, then I wonder if humans can use HRV and resting heart rate kind of as a guide, whether it's their exercise approach or their diet or their body weight, to get that to a more youthful state. And that may be a pro-longevity approach without the pulsatile electric current. So I don't know. What do you guys think?
It's a great idea, but we don't know
Yeah,
if it's true or not. Yeah.
nobody
Mm.
studied it, but your
Yeah.
data suggests that if you can restore the electrical activity of the heart to a more youthful state, it induces systemic rejuvenation, right? So, um,
We believe because because it was a very long-term, effect. We did it once and the effect was, we could see it like a month after or 2 months in those beautiful growth rates, etc. So we believe that we did something to their stem cells, did something, epigenetic changes, something that was maintained for a long time. I think that it's very encouraging to see the comparison to the exercise, and you know we know that exercise has a good effect across the board, right? It's probably not just when we are raising the heart rate, which we are doing during exercise, at least for the time that we are exercising, but it probably, again, improving muscles, improving number of mitochondria in the muscles, has a lot of improvement that maybe the pacing is mimicking it.
Definitely. So you guys cited Michael Levin's work in the paper, and I'm sure you're familiar with his. I don't know. He's got a lot of different published papers and hypotheses and data to support that. But the one I'm really focused on is that he says that, or his data shows that aging is in part because of a loss of cellular goal directedness, and that's being driven by the loss of the bioelectrical signaling, right? So I wonder if when you showed and when you proposed that the pulsatile electric current basically has some exercise mimetic to it, probably the best approach for restoring cellular goal directedness, where cells now, old cells, have to actually work together to, rebuild the body after exercise. So I wonder if that's a part of this story where you're basically giving aged cells a purpose. The PEC is restoring that purpose at some level.
Yeah. Yeah. And this organism is different anyway, in the sense that the body is rebuilt every week completely, except for the stem cells. So there is a major difference in that setting between Batrillus and say vertebrates, mammals.
Yeah,
Um,
but at older ages it breaks down, and then the PEC restores that. So it's almost like
right.
restoring the goal directedness, using the bioelectric signaling to restore goal directedness, thereby slowing aging, which would support Levin's hypothesis. All right, wait. So getting
Mm.
back to the stem cells. So do you guys, I'm sure you guys already have already done these experiments. I don't know if you want to share any details, but how would you set that up? Like, I mean, are you going to have like patch clamped stem cells in order to get electric current into them, or can you do that in cell culture to try to rejuvenate aged stem cells? Yeah.
Good questions. Yeah, we have very, very limited experience so far. We are in a research environment where stem cells are purified all the time, so it's not a big deal to get very highly pure, at least hematopoietic stem cells. And so that is stuff that we'll be looking into in much more detail. But yeah, we are thinking that for some of the initial experiments, you would want to do it in tissue culture, or at least very short-term tissue culture. Whether you can do patch clamp in a bone is a different thing, and that is where you might want to end up. But technically, that's going to be more tricky, but
Yeah, I
doable.
mean, I don't know if you can like barbarian style it and just expose the cell culture to a specific frequency, or rather than because the patch, I don't know. I've got some neurophysiologist friends, and I remember in grad school their distress at trying to patch clamp cells, especially neurons. Maybe it's easier in stem cells, but it's excruciatingly difficult work. Whereas if you could just hit cell culture with a specific pulsatile electric current or even
Mm.
continuous, I wonder if that, that
Yeah, I mean, it works in cell culture. That was actually where I originally used the equipment, is looking at differentiating embryonic stem cells into these contracting cardiomyocyte patches. And you can determine the frequency with which they contract with that machine. Because if you do the in vitro differentiation, each patch contracts at a different rate, because they're not innovated, obviously, and that you can use this machine to change that frequency. So yeah, it does work in tissue culture.
Nice. All right. So I also noticed that there were, although the effects, like when you're looking at the two groups, you can clearly see, I mean, when you run the stats, you can clearly see the shift in the change after PEC for whichever of the phenotypes. I mean, it's like, I don't remember exactly which one. Maybe it was like the zooid size, but there were some in the intervention group that, even though the average of the group is significantly higher, it was a big increase. There were some that were still around the control levels, which suggests that there are responders and non-responders. So have you guys thought about what's behind the non-responders? Like what's holding back the non-responders from experiencing the majority of the phenotype experienced by the rest of the colonies that were rejuvenated?
It's not something we have looked into so far, but yes, it is a good point. How much of it is just biological variability, and how much of it is specifically non-response, and how much of it is measuring artifact. That will be good things to look at in much more detail.
Any ideas what could limit? I get you. I'm not suggesting it's artifact. Don't get me wrong.
Mm.
It's just that
Mhm.
that there were a few that the many experienced the big effect, but there were a few very close to control levels, which suggests that there may be something holding them back from experiencing the full rejuvenative phenotype. And then if you can figure that out, maybe that makes a further boost in the responders. Any
Mm.
ideas, any thoughts of what would hold back? Because they die, right? It's
Well,
not like
one
they
thing
become
could be
immortal.
the way the electrodes are placed would be
Mm,
fairly simple variability between different, if they're kind of on the outside versus on the inside, more whether or not they get the full effect of the current.
that makes sense. And then does Bacillus have a microbiome, like internal?
Yes.
Yeah.
Oh
So
yes,
Yes.
did,
Yes.
did you guys look at
it's a
its
village.
Yes.
Yeah, like
So
all microbiomes, right? It's an eco
yeah.
ecosystem.
So they definitely have microbiome, and I actually we didn't look at it for many years. We were looking on many other stuff. But we do have right now as a postdoc that is an expert in microbiome, and she joined our lab to study the microbiome of Botrytis. And one of the thought was also that maybe these pools simply cleaned the colony or took away, or this is another thing that happened to those colonies, that we actually affected the microbiome while we are affecting other things. And we do have experiment that we run with exposing them to the electricity versus not. First, of course, we needed to characterize their natural microbiome before we can even compare. But I think that now we are in a point that we already know what they have. What are the differences between the wild type versus the one that we are growing in the lab. And again, it's a whole new world that's need our attention.
So something, I'm guessing you're going to see global microbiome shifts in the PEC exposed versus the wild type.
Yeah,
But
I believe
but
so
there
too. But
but there too, it's the intervention is the pulsatile electric current acting on the heart. So if anything, the microbiome effect would be secondary, right? The microbiome is so you rejuvenate the heart pumps more blood, all of the cells and organs in Botrytis are basically rejuvenated to a more youthful state because now they have all.
just that, because you can imagine that we are affecting with this current calcium and
Oh, right. So,
in any cell. Maybe we are opening up gap junctions. Maybe we
yes,
are improving. There's so many things that it can. And this is
definitely.
why we actually need to study and to understand. And maybe it is affecting all of those. And together, this is why we are getting such a robust response.
But what I was suggesting is all of that should improve Botrytis metabolism first, or metabolic functions first. And then so now the microbiome is going to adapt based on the new metabolites that are produced, the more youthful metabolite profile, shifting the microbiome back to a more youthful profile because that's what it's feeding on, right? So along those lines, I wonder, can you extract the microbiome, whether it's
Yes,
yeah, yeah. So
yes,
if
yes, yes.
so if you can, I don't know if you guys have thought about this, but just to propose the idea. So PEC exposed Botrytis, and then after some time, you take the microbiome from PEC exposed Botrytis and transplant that into non-PEC exposed Botrytis. Does that reproduce some of the rejuvenation phenotype without pulsatile electric current, which would suggest that
Mhm.
some of that rejuvenation effect may be mediated, not just a secondary effect of the PEC, some of it may be mediated by the gut microbiome, which would be really interesting.
Mhm.
Yes,
Um,
along the same lines, Botrytis can actually fuse two colonies. So you could do it even at that level. It's obviously something to try that we haven't gotten to, but
So you take the PEC exposed Botrytis that's fused to a non-PEC
and then you
exposed
can
Botrytis.
fuse it to a
and
Oh,
see
non-packet.
for
and
sure.
see the effect.
That was the original interest in the organism. There's a non-self recognition.
But they communicate, right? I'm sure that there's intercellular communication, right?
Yeah,
Mm.
it is. They can fuse with kin. Mother colony can always fuse with its offspring. It's controlled by a single gene, a single locus. If they share both allele, they will fuse and create chimeras. If not, if they share one, also they can fuse and create chimera. But if they don't have any shared allele, it's a polymorphic gene. We call it BHF, the botulinum histocompatibility factor. We did isolate it in 2013, and colonies based on that can fuse blood vessels, create chimeras. In the chimeras, there are competition between stem cells. This is also why we know that it is stem cell mediated. That's also helped us to isolate stem cells because we can lineage trace the originality of each organ in the new developing buds. And there are winners and losers. And this is another part of what we are studying in the lab, to look on what make a winner, what make a loser. And now, looking on the microbiome, there is another level that get into this competition within chimeras. And we have some exciting results on this channel, I guess, but it's still in progress.
Definitely. All right. So you mentioned some of the current and future projects, but are there other current and future projects that you're planning to pursue along these lines?
We definitely could plan to continue with the focus on the nervous system and the microbiome, on the using the pools. We did, in collaboration with the Nakauchi lab in Stanford, we produced about 169 monoclonal antibodies against Butyllus. So that will bring Butyllus to the level where we can actually isolate in a very accurate way, or much better than what we have right now, using flow cytometry. That will allow us to zoom in on which population were affected by these pools or any other experiment that we are doing. We are looking on their immune system. They have a very robust immune system, and we are trying to understand it better. They have many of their unique genes are genes that are associated with their immune system, and this is another place where we are exploring a lot. And again, we have those aging colonies, which is a big advantage. We can always compare between the two.
Nice.
Also,
Does
yeah.
Botrytis have CD4 T cells or something similar to CD4 T cells?
So, as far as we know, in invertebrate, there is no adaptive immune system. Um, it might be there, and it just was changed so much to the level that when we are just comparing on the level of simple sequencing or simple alignments, we cannot find similarities. So we now have some other approaches to look for the way that they are handling viruses and more specific protection. And there are all kind of nice technologies and new algorithms that you can run and look into those problems. So we are implying that, and we are hoping to have some exciting results.
Yeah, it's so far very exciting. So this is great. I think your lab's work is great, and I'm looking forward to more in the future and hopefully having you back on the channel to share those data. So thanks for
We'll
joining
be
and thanks for
happy.
sharing it.
Thank you for
Yeah.
hosting us.
Yes.
Nice
Okay.
meeting you.
Yes, same here. Have a great day.
Okay. Good
For
meeting
you
you.
Bye.
too.
Thank you.
Bye bye.